Hello guys, welcome back to my videos. In this video we are going to see about the modern immunology. Now what is immunology? It is the study of how body protect itself from microbes that cause infectious diseases. So this is the immunology, and microbiology also contain one of the important topic, that is immunology, which is very important to study. So we have seen in our previous video how Louis Pasteur experiment or check the immunization in a particular disease pathogen, and how and he checked out that how infectious disease carried out through the immunization technique we have seen already. And thus after the discovery second time, Eli Machnikov was the person who studied the immunology second time. He discovered the concept of immunology second time. So he was a Russian zoologist and was the first person to recognize the role of certain white blood cells in infection, because infections contain white blood cells. So he was the first person who recognized the white blood cells present in the infections. He found certain types of white blood cells eat disease causing bacteria in animal as well as in humans. Thus he named them as a phagocytes. Now phagocytes means what? The cells which kills bacteria, or the cells which kills the disease causing bacteria. Thus he named the cells as phagocytes, and thus phagocytosis concept was discovered. Now phago means eating and cytosis means cell. So cell eating bacteria or cell eating cells were discovered by Eli Machnikov. For his discovery, he and one person, Paul Ehrlich, received the Nobel Prize in 1908. Paul Ehrlich won the prize for his work on how antibodies are formed. Now, what are the antibodies? Antibodies are nothing but the Y-shaped proteins present in our body and produced by our immune cells to protect our body against the infections. Okay, so Paul Ehrlich was the first person who discovered the antibodies and how they are formed. Later, Burnett introduced clonal selection theory for antibody formation, for which he was awarded Nobel Prize in 1960 along with Peter Medwer. So these two persons, Burnett and Peter Medwer, in 1960 discovered how clonal selection is done in the theory of antibody formation. We will see in later words how antibody formations are done and how clonal selection is done. Then in 1950 and 1960, experiments by Rodney Potter and Gerald Eldman elucidated the basic structure of antibody molecules. So who discovered the structure of antibody? Rodney Potter and Gerald Eldman. These two persons discovered the structure of antibody, and for this investigation they also won a shared Nobel Prize in 1972. Then in 1975, George Kohler and Cesar Milstein developed a technique for continuous production of homogeneous antibodies. So for during the production of antibodies we need homogeneous antibodies, which means similar coding antibodies, you can say. So George Kohler and Cesar Milstein developed the technique by which homogeneous antibodies can be produced. In this technique, you can say this technique was named as hybridoma technology. We will see this technique also in later words, in which antibody forming cells were allowed to fuse with tumor cells to produce the antibodies. This results in the unlimited quantities of homogeneous antibodies, and thus he was also awarded with Nobel Prize in 1984. So you must understand the Nobel laureates. You will need to learn the Nobel laureates name who discovered different types of research or different types of products. Now coming to the interferons. Alec Isaacs and Jean Lindman discovered interferons, which is one of the effector molecule of immune system. Interferons are nothing but the proteins also, you can say, which are very effective molecule in immune systems which is present in our body. Interferons are the substances produced by our body cells in response to viral infection. Whenever we get viral infection or viral disease, our body produces interferons against them. Now coming to the molecular biology and biotechnology, we have seen in our previous lectures also there are different genetics research were carried out by Louis Pasteur, Robert Koch for the immunization experiments. So as a result of these discoveries, in 1940 and 1941, George Biddle and Edward Topham isolated and studied the mutant of fungus Neurospora, which were deficient in their ability to synthesize a particular compound. They awarded Nobel Prize in 1958 for their discovery of genetic material could be transformed from one bacterium to another. So these two persons discovered the genetic material, whatever type of genetic material is there, whether it is in DNA form, RNA form or any chromosomal form, it can be transferred from one bacterium to another bacterium through the experiments of mutant fungus Neurospora. In 1944, Oswald Avery and Colin MacLeod and Macleay McCarthy, while studying pneumococcal disease, found that DNA material from one type of pneumococcus could transfer hereditary characters, that means genetic information, to another type of pneumococcus. That means these three persons, Avery, MacLeod and McCarthy, experiments were confirmed that DNA is a genetic material which can be transferred from one bacteria to another bacteria. So these three persons was the first who discovered DNA as a genetic material. We will see in later words the different types of experiments of DNA isolation or DNA as a genetic material. Then later, James Watson and Francis Crick discovered molecular structure of DNA, from which they awarded Nobel Prize in 1962. So Watson and Crick model of DNA we will also see. Then the study of Jacob and Monod clarified the concept of DNA, which direct the protein synthesis and its regulation. Marshall Nyberg and Herc Mathy and Hargobin Khurana worked to explain the DNA's genetic code. These three persons, Nyberg, Mathy and Khurana, was the first to study or explain the DNA's genetic code, because DNA also contains some type of genetic code. Now these discoveries were established as the genetic information of all organisms contained in DNA. In recent years, major discoveries of genetic processes at molecular level were made using microorganisms. That means in our recent histories or recent past, genetic process at molecular level were made using microorganisms. By using lab animals, genetic processes were carried out. Scientists have also analyzed differential DNA isolated from many organisms. Now, recombinant DNA technology was also discovered at the time, or you can say genetic engineering. These techniques are very useful for generation of industrially important microorganisms which are capable of producing higher yields of desirable products. That means in fermentation or dairy industry, you can see different types of microorganisms helps in producing different types of desirable products. Nowadays many industries have been developed for producing several metabolites also. So these are the topics, or these are the scientists which helped in the discovery of immunology, you can say modern immunology techniques. Thanks for watching.